Okay. Started off weak, but got better as time went on.
That's how we go. That's how I go.
That's how you go.
Yeah.
Hello and welcome back to the Brainfart podcast. 3?
Yeah.
Oh, 2, 3. Okay. I'm Sean, as always. That is Matt, as always. We're always the same people. Lockdown update.
Oh,
That's become relatively customary in the last few episodes.
oh, okay.
More or less since the coronavirus one.
Yeah, we have more freedom now.
And non-essential shops are to reopen a week from the day of this episode uploading.
Ah, okay.
So this will upload on the 8th of June and shops will open on the 15th.
That is, yeah, I did already know that actually.
So on the day that the last of this block of 3 episodes is uploaded, people will be able to go to like Primark and buy clothes and stuff.
Yeah, until the R increases and everyone has to go back inside.
Yeah, and more people die.
You know, the R's, I saw some data that show
Has it already started?
estimate that it's most of the country, it's like 0.8, 0.9 at the minute. So
we're basically almost at the point.
Back
so
inside,
they're
everyone.
literally going to open it just in time for them to go, okay, everyone.
I think that's what's going to happen.
Do you agree with the notion that Boris is pushed or rushed the reopening of stuff to try and stop people getting so pissed off about the Dominic Cummings situation?
I think a lot of this stuff had been published before that, or maybe it was published right around the same time.
Right.
And to me, if they were published right around the same time,
then
that
there
seems
wasn't
a
enough
very
time for
instant reaction to have like a plan ready to go with dates and stuff.
Right.
But maybe it's certainly possible.
Yeah,
It's
I wouldn't put
possible.
it past him. The only other
BJ
Boris Johnson, big BJ. The only other piece of news, and this is going to be a little sort of secretive, is we're going to be implementing a few interesting changes in the near to mid future.
Operation Code Red.
Is that what we're calling it?
It is now.
Okay, you've chosen the code.
Yeah.
The code name. Beautiful.
The Operation Code name Red.
Yeah. So last time, what did we do?
We talked about cells.
We did, and what they are.
We talked about
And what they do, what they need to be do.
What cells need to be good cells.
Mm-hmm.
So we've got this picture of our bubble filled with
bubble cell.
Yes, bubble cell.
Bubble-like cell.
Bubble-like cell. Just imagine a bubble filled with cell juice.
Lots of lovely cell juice, cytoplasm.
And other sort of mini bubbles that we call organelles that we talked a little bit about.
Mitochondria, ribosomes, little things like that.
Those are the organelles.
Yep.
They do things inside the cell.
Yep, they do.
So we talked about what cell has to do to be good at its job.
Mm-hmm.
What different thing, how that relates to what like a whole organism has to do.
We talked about Mrs. Gren. Give us a little recap. Can you do it? Can he do it?
Movement, respiration, sensors, growth, reproduction, energy and nutrition.
Nice.
Excretion and nutrition.
Yeah.
Because energy and nutrition are the same thing. Yeah. So, yeah, we know what a cell has to do to exist and be successful.
And why cells are important.
At its job. Before we come on to what we're going to be talking about this week, I would like to talk a little bit more about the episode last time.
We have a few amendments.
Corrections.
Yeah.
So, a plasmid is actually not a bacterial genome, at least not in its entirety. The main bacterial genome, it does exist as a circular piece of DNA, but it is not the plasmid. The plasmid is considered extra chromosomal DNA.
Right.
So DNA that is not part of an actual chromosome.
Okay.
I actually didn't know that. Whether it's because I didn't pay attention in lectures, or whether it's because it's. Is
it a bit of a slightly obscure thing? Yeah.
It's not that obscure, but when we talk about applications of genetics and things like that, plasmid is a much more common use. In genetics, we use them a lot in genetic engineering.
Well, in any level of education, things are slightly dumbed down to what they actually are. Even way up to like master's level, they'll be dumbed down compared to what you'd learn at a PhD and stuff like that. And at secondary school, it's dumbed down from A level and etc.
Masters is pretty good. Degree, graduate study, and undergraduate study and graduate study are pretty good. It's the different stages in academia are more like, at that point, it's like breadth and depth of knowledge.
Right.
So it's like, how deep can you get? Because there's so many like different biological molecules and stuff that can interact with one another.
Yeah, and it keeps going and keeps going
Exactly.
down the rabbit hole.
Exactly. And then you've got like physical interactions, molecular interactions with sizes and stuff like that.
And then
like it gets
quantum
crazy.
interactions when it gets absolutely mental.
Maybe. So some bacteria, however, do have a linear genome as well.
Right.
And I was talking about genetic engineering. The thing that I thought was called a COVID is actually called a cosmid, and it's not a type of bacterial genome. It's like a collection of genes that have been put into this plasmid with this kind of sequence from a kind of virus. It gets a bit complicated, but basically they're used as kind of stores almost of genetic information.
Right.
So rather than having like a load of individual genes or something stored somewhere on ice or whatever, you'd have one cosmid with a lot of genes on it, and you could easily then just use those genes for whatever it is you wanted to use them for. Or you could transfect them into a bacteria to use them for example.
So a cosmid is a plasmid with extra stuff, essentially.
Yes, it's a cosmid that's basically
It's a plasmid.
it's a plasmid. Sorry, that's basically been had stuff put into it either for storage or to make it behave in a certain way.
Right.
Mostly it's the way I've heard them being used is as storage forms of genetic different genes that we want to use to transfect into bacteria.
Okay, sure.
So genetic engineering, great place to segue into what we're going to be talking today. What we're going to be talking today. I'm still doing it.
It's never going to end.
About today.
It's never going to end. I'm going to just make a compilation of all the times you mess up.
I'm down. So genetics. So I thought, again, we'd start with a little question.
Go.
What is DNA?
Well, I can tell you what it stands for.
Go for it. Anything you want to share.
Deoxyribonucleic acid.
Mm-hmm.
And it's essentially, as we discussed, sort of touched on a little bit last time, it sort of contains this genetic information, and that genetic information serves as instruction for cells to do things and the body in general to do things.
Yeah, exactly. So pretty much that's right. I'll build on a little bit. DNA is an information storage molecule.
Right.
So in the same way that a computer has a hard drive for its memory,
mm-hmm.
and those are all stored as ones and zeros, the cell has its own little storage area.
Are they ones and zeros as well?
No, they are base pairs.
Which TAGCU.
Yes, use an RNA, which we'll come on to. But basically, it's a fairly complicated molecule, but basically all you have to think about is they pair up in these A's and T's, C's and G's.
Yeah.
And they line up along a line, and it's just a long stream of these codes, these bases, nitrogenous bases.
T is always with A, G is always with C.
Yes.
So you could have TAGC, ATCG.
Yeah.
Yeah.
If you were looking across.
Yeah.
So exactly. So it contains within it all the information for producing all the little molecular tools that a cell uses to accomplish its missions.
Mm-hmm.
So those are proteins, basically. So these are actually how the cell does all the things it needs to do. So anytime we were talking about like communication, that's all accomplished through receptors and then chemicals that bind to those receptors. We were talking about things like grip proteins for the cell adhesion.
Yes.
The cells stick together so that they know whether they're with other cells.
The little almost zip.
Yeah.
Like.
So they're proteins. They're proteins, and the information for them is stored in there.
For them to be created. For those proteins to be created.
Is in the DNA. So it's not just muscle that we need protein for. It's basically everything that the body does, everything that an individual cell does. So that is the reason why DNA and genetics is important, because it basically explains to you how the cell can build the things that it uses to do everything.
Yeah, so last time we obviously talked about why cells are important, how important they are.
Exactly.
But in order to have those cells do those important functions that we need to happen, we need that DNA to give those instructions to these cells to create these proteins to act out all these functions, right?
Exactly. And not only that, but then looking at the genetic code, the DNA code, because that's then used and read in a certain way, which we'll come on to create these proteins. You can look at the genetic code and get some idea about what these tools, these proteins might look like and how they might behave. And that's obviously, studying that is the basis behind looking at mutations and things like that, because any differences between different people's genetic code will be because of mutations one way or another. So the genetics can tell you a lot about different ways that the cell might be impacted by things like that. The basis of human genetics at least, yes, is something we call the central dogma.
Okay, I've heard you say that before.
You've heard about this a little bit.
I wouldn't be able to tell you what it is.
So the central dogma isn't really a dogma as such, because basically a dogma is something that you sort of blindly follow without evidence. So like religion.
Right. So it's just sort of the way it is, sort of thing.
Exactly. However, the central dogma has been obviously probed and tested, given that it's science. So while it started as a dogma,
it is now ingrained in fact and evidence.
Exactly. It looks like really that's the way that's the way it happens.
Mm-hmm.
So the central dogma is basically, you start in this this nucleus with the DNA, and then with the DNA you make RNA, and that's called transcription, so writing down, and then from that RNA you use to make proteins, and that's called translation. So you transcribe DNA into RNA, you write the code down in a certain way, and we'll talk about why that's important.
Then you take that written down code
and you translate it into a protein.
Okay.
And then the protein can go off and do the cell's bidding. The
whatever function needs to be acted out.
Exactly. So the reason that we don't go straight from DNA to protein, there's a couple reasons. So the first one is that there's actually a lot of DNA that doesn't code for proteins. A lot of DNA. A hell of a lot of DNA that does nothing.
There's a name for it, isn't it? Sort of innate.
Junk DNA, non-coding DNA.
I think that's what I've heard, non-coding DNA.
Yeah, so
it's sort of innate. It's just sort of there. It serves no direct purpose, but it is part of the DNA code itself.
There's what it looks like, and is that a lot of this DNA that we thought didn't have a purpose, like I say, we used to call it junk DNA. Really what it looks like it's probably doing is having some kind of regulatory function on other aspects. So we were talking about this might be a conversation I was having with someone else. Were we talking about viruses and how they evolve at any point?
I believe we have at some point. I don't know whether it's the conversation.
Basically the point is you could think of a mechanism, a way that it might be beneficial for organisms to be able to control the rate of their evolution, for example.
Right.
Genes and the non-coding genes might have some kind of biofeedback mechanism for reading what the environment's saying.
So, for example, it might have something to do with, you often hear scientists talk about how our evolution as a species, as the human species, has slowed down dramatically, for example. So you're saying that could have something to do with regulating, we don't need new genetic mutations because we're pretty suited to the environment we live in right now.
Maybe, yeah.
Right.
I'm saying that's one potential explanation for this junk DNA. Other things are, obviously biology is a really finely tuned and well moderated system. It has to be. And the vast amount of that junk DNA that we used to call it junk DNA, now we call it mostly non-coding, or introns, another word for them as well.
Introns.
We'll come to that.
Right.
So the vast amount of non-coding DNA probably points to the fact that obviously if you need really tight regulation, you're going to need a lot of information to dictate how things will be.
Everything's regulated.
Exactly. So first reason we don't go straight from DNA to protein is there's so much non-coding DNA that doesn't really need to be turned into protein. It has some kind of other mechanism of action.
Could you call it sort of ambient DNA in the sense that it could serve this ambient function that you can't see on a grand scale, but it might also not necessarily serve any function?
Yes. So for example, there's a lot of sections of DNA that are just left over from viral infections. And they could do things, they could be beneficial to the organism in some way, but at the same time, they might just sit there and be really good at staying on the gene.
Clinging to it. Yeah.
Exactly.
Gotcha.
Exactly. So the second reason is, don't go straight from DNA to proteins, is that all the machinery, the molecular machinery that we use to make proteins exists in the cytoplasm.
Yes.
And because of the conditions within the cytoplasm, if you exported the DNA from the nucleus into the cytoplasm, you would likely get DNA damage, and then you wouldn't even be able to get the protein because the DNA would just be broken down.
It's like a computer code where you delete a load of the code, and it can't do that function, right? Is it ribosomes that make the proteins from the RNA?
Yes.
Okay. I just wanted to.
So the two, we'll talk about the enzymes. I think I'm about to come on to how they work, at least at a somewhat vague level. Mm-hmm. So we won't talk necessarily about exactly how the process works, but just how to think about what's going on. Mm-hmm. It's from DNA to RNA. So in transcription, that's called RNA polymerase.
Yes.
Yep. And in going from RNA to protein, translation, that's a ribosome. And tRNAs or transfer RNAs.
Yeah, I always heard it as transport RNA.
Oh, I've never heard that before. Okay.
Maybe I'm misremembering then.
We work with different academics, so it could easily be a way of calling it. There's loads of different things for loads of different things. So we'll talk about transcription first. So like I said, because the environment of the cytoplasm is going to be damaging to the DNA. That happens in the nucleus. So basically what happens is
this the thing, you're obviously about to explain it, but the unzipping process.
So this does happen during, So you have this RNA polymerase causes the there's another enzyme that causes that. It's called helicase.
That's it. Yeah.
So helicase causes the unzipping of the, and it's an unwinding as well. So the helix actually gets straightened out.
Yeah. So you imagine this. I mean, it's hard to do with your fingers, but it's all spiraling round. It's unspiraled, split down the middle.
Yes.
Along is it hydrogen bonds that hold them together?
Yeah.
I'm impressed. I'm remembering this, to be honest. And then it copies one half of that one of the strands, right?
Almost, so it doesn't copy it, but in its process, it might as well be copying it because it doesn't copy it. It just writes the strand that's opposite to the opposite strand.
Yeah, so it copies it in the sense that you're writing down what would go with that strand. Yeah.
Yes. And obviously, at that point in RNAs, you replace the T's, the T's in the code with U's for uracil. Just realised we haven't talked about the double helix yet. So structurally, DNA exists as this coiled up, wound up.
Everyone's obviously seen this double helix.
DNA looks like.
Everyone will know what that is.
So helicase starts the process, unwinds it, and then you have this RNA polymerase binds to the unwound area. Now the area it binds to is called a promoter region, and this is. Understanding this sort of terminology is super important if you want to look at genetics, because these two terms that I'm going to come on to now, if you look at how genes are read, basically this is everything. So this defines where the gene is.
Okay.
In fact, just think of it. Do you know what the definition of a gene is?
No, I don't think
so.
I do, actually.
So it's quite hazy, actually.
What, like
gene,
indistinct sort of thing?
yes.
Oh, okay.
So a gene is just any length of genetic material. That's pretty much what it is.
And it has to be at least two lengths on the DNA, isn't it? Two or more base pairs to code for.
I would say it has to be functional in some way.
Rather than it being the non-functional.
It can't just be a piece of DNA that's not doing anything.
Okay.
But because like genes will overlap, so let's say you have this one protein being coded for here, you might go not all the way to the end of that gene, and find another gene for something else.
Mm-hmm. Sure. So genes would make up a genotype, and then that genotype would then manifest itself in a phenotype, for example, right?
Yeah.
Without it then eventually becoming or resulting in a phenotype, it can't really be a gene because it's not serving any purpose. That's sort of what you're saying, isn't it?
Yeah.
It needs to have a result in order to be considered an actual gene.
Yes, I would say so. You can get loose or tighter with that definition if you want to. It depends on what it is you're looking at in either research or discussing.
Yeah, I suppose it will change depending on what area of genetics you're looking at and what it relates to specifically.
Yeah, exactly. So, yeah, you get this helicase which unwinds. RNA polymerase comes and binds to this region that we call a promoter region. So a promoter region is just a fancy word for meaning start here.
Okay, that makes sense based on the name, I guess.
Bind to the thing and then start reading.
Mm-hmm.
And it causes like you were saying, the code that it reads on the DNA, it then matches up the bases, like A's to T's or U's in this case, and C's to G's, and matches up a strand along one of the sides of the DNA. Mm-hmm. And then eventually it reaches what we call a terminator region, or a terminator.
That was actually impressive.
Yeah.
So presumably that just says stop here.
Yep, that's all it means. It means stop here.
So the promoter region, start here, terminator region,
stop here.
Stop here. Okay,
yes.
Gotcha.
And what happens then is the RNA polymerase will just fall off the DNA. The mRNA will also just fall off the DNA. And we were talking about these coding versus non-coding regions of the DNA. And we were talking about introns and exons. I didn't mention exons yet, but basically, when you have the DNA, any area that is coding for something is used in the actual coding sequence, we call it.
Okay. That's
called an exon.
Right.
However, if it's not in the coding sequence, we call that an intron, because that doesn't actually have a massive effect on the resulting protein.
Okay.
It might have regulatory effects, but it's not being read by the ribosome.
It's not part of the main code.
Exactly.
Yeah.
So what will happen? We have this thing called the spliceosome, which cuts all the introns out.
I've actually never heard of that, I don't think.
Spliceosome.
Yeah.
Just like a magic pair of scissors.
Nice.
And it cuts all the introns, all the non-coding regions of that DNA out, and then that mRNA is exported out of the nucleus.
Mm-hmm.
So
into the cytoplasm.
With only its exons intact. Obviously, at this point, we've gone from our double-stranded alpha helix DNA to our single-stranded, probably still interacting with itself and curled up in a ball somehow.
Yeah.
But single-stranded RNA. So this gets transported out of the nucleus, like I said, and then we come to translation, which is
ribosomes.
Yes. And really, it's pretty much the same concept again, but you're not having anything. Rather than it being the DNA bases that you're lining up, it's a new code, and that's why it's called translation.
Mm-hmm.
So we're going from RNA to protein, and that happens in the cytoplasm, like we've said. The main a big reason for the RNA is that the RNA doesn't degrade as easily in the cytoplasm as DNA does. So what happens is this transfer RNA comes along with an amino acid attached to it. And proteins are made up of amino acids. So basically the transfer RNA or the transport RNA, if that's what you've heard, is basically like a shuttle for the amino acids to come into the ribosome to get checked to see whether they match up with the codon, we call it, on the RNA. So a codon is just a section of 3 bases, and effectively the transfer RNA will have what we call an anticodon on it, which is those 3 bases but the opposite base.
Ones.
Yep.
Yeah.
So effectively what happens is the transfer RNAs come into the ribosome. The ribosome effectively like checks whether they match up.
Basically making sure everything's the way it should be,
Yes.
right? And if it isn't, then does it get rid of it? Or
Yeah, it just gets rid of the tRNA will just be ejected from the ribosome.
And presumably then they go to lysosomes to be excreted.
Yeah, or they can be reused.
Okay,
they can just,
right.
Yeah.
Oh, that's interesting. I didn't know that they could be reused. Right.
Everything. So everything in biology, when you're talking about like molecular events,
if it can be reused, it will be reused because you want to waste as less energy as possible.
Exactly. So you get this: the ribosome binds, the tRNA comes in, the tRNA and the ribosome check whether this codon matches the anticodon on the tRNA, and if it does, then it'll grab the amino acid and say, just stick this on the together. End. Exactly. Just stick.
Yes.
Just stick this on the end.
Yeah.
So the way I think about this is the ribosome is kind of like a quality controller.
Yeah.
So it's the guy.
So man on the end of the conveyor belt going, yep. Nope. Yep.
Exactly. So, we were talking about promoters and terminators. So the language is a little bit different here.
Okay.
What we have obviously codons. So we have a start codon and a stop codon.
Okay, that's easier to understand, I guess.
Or sometimes they will be called terminator codons.
Oh, okay.
Stuff like that. But there's actually 4 of these codons. So there's 1 start codon and 3 stop codons. And the 3 stop codons are actually like traffic lights. So we call them...
Oh. Yeah. Why would that be necessary?
So we were talking about how genes overlap.
Is it about
sometimes.
Speed or something?
Kind of. So we're talking about sometimes how genes overlap.
Yeah.
Let's say maybe you have a version of a gene that might be smaller than, and it does a slightly different job, but it's effectively the same gene.
Thing. Mm-hmm.
It's just cut off a bit early. Right. If you have, say, a green light, then maybe most of the time that gene gets read all the way to the end. And you don't get the smaller form of the protein.
Mm-hmm.
Let's say if you have a yellow light, then more, say it's closer to 50-50. So you get about an even proportion of the smaller protein and the bigger protein.
Yeah.
And if it's a red light, let's say most of the time it stops early. So you get a greater proportion of the smaller protein and much less of the larger protein.
Yeah.
So it's effectively just another regulatory mechanism.
Uh-huh.
So all of these things, the things that seem like, oh, why is that? To me, most of the time it seems to be like, oh, this is just another way of fine-tuning the dials, another way of just making sure you get all of the right things and not too much of the right things and not enough of the right things.
Mm-hmm. Just having as many subsystems to be checking everything as possible to know that everything is as close to perfect as it can possibly be, sort of thing.
Exactly. I mean, it's like we were talking about with the cancer in the cell cycle. So constantly cells are checking what's going on in so many different ways. Again, that's why I think probably there's so much junk DNA, is because there's so much regulation that needs to go into making these things.
Because everything's so complex.
Exactly. So complex. So you reach termination codon in the process of translation, and then again the ribosome dissociates, the mRNA dissociates to then be destroyed, and you have your protein then.
And that can be taken to... That's where the endoplasmic reticulum and everything comes in for transportation. Obviously, that's not really what we're talking about here, but that's the next step would be. So
basically, at that point, you've got your protein. Basically, what proteins do is they have areas of themselves which will interact with other areas of themselves, and they basically fold up into a squiggle.
Yes,
That's how we
I can remember
draw
when
them.
I was doing that assignment, we saw that photo where it's just this folded up on top of each other and all this big mess of strandy goodness.
Exactly. And the intramolecular forces in the protein, they then result in whatever function it has. So the dynamics of how that protein interacts with itself and the other molecules around it, that dictates what the protein will do and how it will be used. That's getting a little bit, basically it's chemistry.
Okay. Right.
Effectively, the way I always think about it is like they're kind of like, they are just little biological machines. In fact, there's a lot of work been done in the last sort of 5-10 years to make biological machines out of proteins. There's a really cool video of these chemists who have built a system with a central molecule that has 4 molecules on sort of the corner extremes. And basically the central molecule is supposed to be like a chassis of a car, and the outer molecules are supposed to be wheels.
Oh, okay.
So you can see this thing trying to move along it's a microscope
That's
video.
Cool. Do you have any idea what people could search to find that? Because that does, I feel like you might have actually shown me that, but for anyone watching that might want to try and find it.
I would try something like molecular machines car, something like that.
Right.
That's what a lot of it won a Nobel Prize actually a couple of years back, 3-4 years back, something like that.
Do they do a Nobel Prize every year?
Yeah.
But there's different types of Nobel Prizes, isn't there? Yeah, I don't really know much about that.
Yeah, neither do I.
You're a scientist, you should. Maybe you'll get one day. Who knows?
So the point is that proteins fold up, and then they do the function. Like you're saying, you have the endoplasmic reticulum. You also have the Golgi apparatus.
Yes. So it goes. Proteins go through the endoplasmic reticulum. They're transported to the Golgi body, and the Golgi body is basically a packaging location, right? Everything's sort of packaged up in a neat little transferable or transportable vehicle almost, or package, depending on how you want to look at it, and it can be transported to different parts of that cell, to different cells.
Yeah.
However, wherever it needs to go, basically.
Yeah, either they can be packaged if they want to be exported from the cell, or
do they only really need to be packaged if they're going to be?
It depends. So maybe they need to be transported to maybe they're membrane proteins.
Yes.
Maybe they're receptors, stuff like that. Then they'd probably be packaged then.
So anything that's either sort of border cellular or extracellular, it would presumably be packaged. Anything that's directly intracellular
might not be.
Okay.
Yeah. But many cells do travel still to these organelles because there need to be many cells. Many proteins do still travel to these organelles because they need to be edited in some way.
Okay.
And when we talk about protein editing, effectively all it is.
Well, that's them being edited after they've been created. Yes. I didn't even know that could be done, to be honest. I thought is that in case something's been missed as it's been transcribed in that ribosome, or is that sort of like a post thing where you go, okay, that was correct when we made it in the ribosome, but we need to change this because it needs to now serve this different function?
It's another regulatory mechanism.
Okay.
So basically you make your protein, and your protein is kind of like a machine, but just the mechanics of it, right?
Okay.
So let's say you've got this machine and it's got a load of switches that change like the speed that it works at, and maybe other things like that, how efficient it might be, things like that. So you have your protein made, and then it will go to these organelles, and effectively all you do is you stick different types of molecules on. So you might have heard of phosphorylation before.
I've heard of phosphorus, but not phosphorylation.
Yeah, okay. So
is that just putting phosphorus?
The addition of a phosphate group.
Right.
So like this, basically
a phosphate molecule is thrown onto it.
Yeah.
Would that always change it in the same way, in a consistent way?
Most of the time, phosphorylation causes more active proteins. So generally speaking, when we talk about phosphorylation, that will increase the activity of a protein.
So it makes it do the thing it's supposed to do more often?
Yes.
Okay.
However, it does get very complicated. There's things like we call glycosylation, palmitoylation, some even longer ones that I can't say without having them in front of me. So there's lots of different ways that proteins can be edited, and all that does is it changes the way they work in some way. Right. So that's about as specific as you can get with it, because it depends on, I was saying the forces between the molecule of the protein interacting with itself. It makes changes to those forces by adding other molecules onto
to interact with that sort of tiny physical interaction.
Exactly, and that's literally how a protein works. It's these molecular forces that are causing these more sort of almost pseudo-physical interactions. So the point is that when the protein's done, it can be fine-tuned by some of these organelles, and there's a few of those organelles that do that job. So one thing that we've not talked about is mutations in genetic disorders in any detail so far.
Yeah.
So I wanted to come on to that a little bit, because obviously mutations are a massive part of both genetics and evolution, and that's what we're trying to.
And I feel like mutations is the sort of thing that most people think about.
Right.
When they think about genetics, right? Certainly, it's the main thing I sort of go to.
The X-Men.
And yeah, exactly right. The fact that everyone thinks about genetics and how that affects your eye color and your hair color, and how blue eyes is a mutation, stuff like that. So I feel like that'll be quite approachable for a lot of people.
Yeah. So effectively, all a mutation is we have what we call a reference genome, right? So that's basically just kind of like they're obtained in different ways, but it's kind of like an average of the wild individuals. So you go out into the wild and you say, obviously when we talk about wild, normally that would be like animals.
Yeah.
But you go and you however you get it, and it's supposed to be like an average genome. So you say, this is what the average mouse looks like.
Genotypical?
Yes.
For example.
Yeah. So like, exactly. So genotypical, like the what you'd expect to find. And basically, if you then look at someone else's genome, you can see all the differences between the reference genome.
What has changed
exactly
from this genotypical
all of those changes are mutations.
Yes.
And it just changes to the code, the DNA code. So where you had an A at one point, you might have a G instead, or it might be replaced with a T, so that it's swapped over sort of from Yep.
And I mentioned, just to quickly mention, I was talking about genotypes and phenotypes, but I don't think I specify what the difference is between those two.
No, you didn't.
So genotypes is the code essentially, isn't it? And the phenotype is the physical attribute that results from that code, right?
Yeah. So a good way of thinking about it is so if you're talking about eye color, because I think that's one that a lot of people have some kind of knowledge. Yeah. So people know like big B, little b for blue eyes and brown eyes. Yeah. You might have big B would be the genotype, and that would produce the phenotype of brown eyes.
Yes.
Yeah.
Go on.
So mutations, they're changes to this code. So you would have changes to the genotype that cause effects in the phenotype. And basically, when we think about what these changes can cause, effectively there's more codons for each amino acid than there are amino acids. So for each codon, for each amino acid, we have multiple codons that can say, put this amino acid there.
Okay.
So because of that, you can actually have mutations that do nothing, that change the codon of the RNA to make it the same amino acid.
Okay, sure.
Right?
Sure.
We call those synonymous mutations.
Right.
They can have, again, sort of these small regulatory kind of effects.
That's the word of the day, regulatory.
Yeah, regulatory. Everything is, the most important thing in biology is regulation.
Yeah.
But they don't tend to have, generally speaking, massive sort of game-changing impacts on an organism. And then you can have a something that will change the meaning of the codon. So it will change an amino acid. And we call those non-synonymous mutations.
So they're ones that actually impact something or other.
They might not necessarily impact things in a big way, but they will change an amino acid. They will change the protein's code.
Okay.
And that's why they're non-synonymous, because where you did have a codon is kind of like a word, right?
Mm-hmm.
Where you did have the same word there before, but just spelled a bit different.
Now you have a different.
Now you've got a completely different word. So these non-synonymous mutations, because they change the amino acid sequence, we won't dive too much into like protein structure, but effectively there are these 20 amino acids, and some of them behave in chemically similar ways. So let's say you have 2 amino acids being swapped that are chemically quite similar. You're less likely to have a massive difference.
Impact.
Exactly.
Right, yeah.
Whereas if they're completely different, then you're likely to have more of an impact on the protein's function.
Yeah.
Exactly. And not only this, what we can also have, we call them missense mutations. So this would be when you've got something that completely changes the how the how the RNA is read, how the DNA is read. So for example, let's say you inserted an extra base pair. So that would shift everything over one. And because you've got these three letter words, that changes the spelling of every word. Right? So imagine if you were reading a book and all of a sudden there was an extra letter, but the end letter of every word went into the next word.
Yeah,
that's what it's like. So it completely changes
the context of everything within
exactly.
the code, right?
So when things like that happen, you're much more likely to get something that will completely destroy the way the protein works, because maybe you've put a stop codon way earlier in the RNA sequence than you normally would have had one.
So you might have an entire missing part of
exactly,
that. Right.
Exactly. Or maybe the protein just doesn't really get made at all. Maybe you get rid of the start codon.
Right.
So a lot of those kinds of major things, especially when they happen to really important proteins, and we'll come to talk about that in a minute, especially when they happen to really important proteins, they tend to be lethal quite early. Some of them can be. So this is what I'm actually coming on to talk about now. So what kind of mutations will propagate versus won't propagate? That's the answer. Is if it's mutations to a really key process, so like we were talking about mitochondrial mutations or other energy systems, so any of the enzymes that the mitochondria uses to produce energy, if they're mutated, you're gonna see quite early death.
Yeah.
Things like proteins that contribute to brain development, potentially if you have mutations in them, you're going to see quite an early death. So if you see quite an early death, the organism is far less likely to reproduce, which means in the future you won't see that mutation again.
Right. So the worst mutations by definition aren't around very long.
Yes.
Because they can't. The people who have those mutations aren't going to survive long enough to reproduce.
Yeah.
Okay.
And that's why you don't see that many people with these like really crazy genetic disorders, because they mostly have to be spontaneous mutations, so they can't be hereditary, so passed along from parent to. They can be in some sense with things like we won't get too much into heredity, but things like you can be a carrier of a disease like haemophilia, for example.
Yes, that's the main one I like.
Yeah. So the point is you have to think about how that might affect the organism's ability to reproduce, and then that will tell you how likely a mutation is to spread throughout the.
And that's why I suppose you see the sort of most common brain affecting mutations are sort of things that include like learning difficulties and things like that, because that's not going to affect the organism's ability to reproduce.
It's not massively going to affect it. Exactly.
Right.
Exactly. And actually, great segue into one of the sort of the last. We're drawing to a close now. So you're talking about, that's why you see brain disorders and learning difficulties being one of the more common forms of mutations that you see.
Yeah.
Um, one interesting thing about genetics is this thing we call pleiotropy. So pleiotropy is basically the idea that certain genes might have different effects at different stages in life.
Okay.
So the genes that have beneficial effects early in life are much more likely to be selected for because they help the organism reproduce.
Mm-hmm.
Whereas the genes that cause, are good for you later in life are less likely to be selected for, because whether you reproduce or not,
it's kind of irrelevant because you've
already there.
You've
already reproduced by that point.
Yeah.
So basically, you think about the kind of things that really impact. Go on.
And is that why, as you get older, the more these negative genes that result in dementia and things like that happen? Because if you're going to have reproduced, you have at that point.
Yeah.
So the body doesn't need to focus on avoiding those more harmful genes.
Exactly.
Okay.
So that is exactly what it is. All of these genetic factors behind different dementias and things like that, they can't be selected against, because the organism, when generally speaking, there are some like early onset Parkinson's and stuff like that.
Yeah.
The organism has already reproduced by the time the onset of the disease. So there's no inherent way other than like sterilizing people, which don't sterilize people.
Yeah, no.
Other than doing things like that, there's no inherent way to prevent those being passed on.
Yeah.
So you see a vast number of genes being implicated in dementia type disorders, neurological disorders that do that. And that's all kind of this pleiotropy thing, where something might be really good for you early in life, but then over time, the effects of that might contribute to then producing some of the pathologies that we see associated with old age.
Yeah. Because we've obviously talked about how that's really all genes want to do, is reproduce healthily and keep their own genes going.
Yeah.
As long as they can.
Yeah.
The whole concept of meme theory and stuff like that.
Yeah. Well, gene theory. Gene theory. Yeah.
Meme theory. We tried.
But yeah, exactly. So the idea is just the gene wants to carry itself on. If the gene can reproduce, that's all that matters.
Yeah.
All that matters is I am in the next generation of
I keep going.
Organisms.
Yeah.
Exactly. So the last thing I wanted to come on to, now we're talking about mutations. Not only can you have these, we've been focusing more on sort of hereditary mutations, so things that you would pass onto your children,
like colour blindness or haemophilia, like you mentioned.
Yeah. But you can also have these somatic mutations, so mutations within the body cells, and that's obviously what causes cancer.
Yes.
Because you it's not like, oh, you had a cell that was cancerous and it was just waiting for the opportunity to strike. No, you have the DNA damage that causes rewriting of the genes, some of the genes, and that then produces cancers. So I won't go too far into it, but basically the idea is that both mutations and assortment exist to create a space where evolution can be explored, and they also exist as accidents which cause the cancer. And again, there's a whole host of different hallmarks of cancer. So I think there is 11 now, and the different mutations in different genes and things like that will cause the different cancers. But the key point there is that normal cells can have mutations. Most of the time, that's a cancerous growth.
Right.
I think that's where we'll leave it for now.
Okay, beautiful.
Next time we're gonna dive into something very dear to my own heart.
Yeah,
which
it's
is
very, very close to your interest. Closest. This will be the closest thing we've talked about on the podcast to what you're really into, isn't it?
Yeah, and it'll round out the. I think with these 3 episodes, this one, the previous one, and the next one, it should, for people who want to understand and have some inherent idea and analogous ideas and be able to visualize what's going on in biology and have some way of deciding whether or not what people are saying is possible or true. Or I think with these, it does a great job of starting. The only area of the like biological systems that I think these three episodes don't cover are electrical activity in the brain. That's literally it.
And that's something we'd go on to talk about eventually anyway, sort of thing.
And really all that is wiring and stuff like that.
Yeah, I feel like the best way for anyone who is interested in using this sort of sub-series almost would be to watch them and then go and watch more directly educational videos with visuals and everything like that, right? So you'd sort of watch these 3 episodes, you'd pick up on some stuff but not all the stuff, and then you'd watch some other videos, some more visual stuff, and you'd it's like we've spoken about this before, how we'd discuss things in biology and I wouldn't quite get it, and you'd sort of say, yeah, but next time you hear that, because you've heard it before, it's going to stick a lot easier, right? Yeah. So people would then watch these more visual videos and or podcasts, whatever they want to do, and things hopefully would stick a bit easier than if they'd just gone to that content initially.
Yeah, and I think certainly that's the way around to do it. Yeah. Like, so for me, even.
And I suppose that's why, just to interject, podcasts are so good, because it's the whole point is ambient learning. So you can sit there with it, even half listening to it, and then go on to do the other stuff.
Yeah, I think even when you have top quality teachers who are instructing you through this, going at it academically is quite intensive and it takes a lot of like processing power. Whereas just sort of discussing kind of what's going on is much easier. And if you understand sort of colloquially what's going on, so then you can see what's going on and go, oh, rather than being confused, I have some idea that's probably a protein that's doing something. And, what is it doing? Oh, we haven't talked about what proteins do yet, so I'm not sure. We'll have to watch more Brainfart podcasts.
Yeah, nice. Really nice.
Shameless plug.
So thank you for watching. As I said earlier, we've got a cheeky plan, more of a plan than we've ever had, really. It feels like we're going full steam ahead, more so than we have in the last year or so. Another bit of news that I forgot to mention at the start of the episode is the website. Within the first few episodes that we made, I made the Brainfart blog, which was designed terribly, and we made one post and then just forgot about it. But now we have a more professionally designed, it's quite beautiful website. So that's where you're going to get news and more updates, as well as some exclusive clips and stuff like that will be on there for people.
Exclusives.
And that's where people will hear the first of our soon to be implemented 16-stage Code Red Code Red plan.
Operation Code Red.
Is that what you called it?
Yeah, it was close to that.
Like that. Yeah.
Instigate Operation Code Red. Yeah. Something like that.
Yeah. So thank you again for watching, and we shall see you. Get out my shot. Yeah, that's what I thought. Thank you for watching, and we will see you next week, where we're going to look at neurons and neurotransmitters, things like that, right?
Cellular communication.
Cell
signaling and neurotransmitters. And
communication.
Not neurons, though, because that gets a bit complicated.
Cellular
communication.